Good morning, everyone. I'm Priyanka Ghosh. I'm an acting team lead within the Office of Research and Standards, Office of Generic Drugs. And following Dr. Wang's presentation today, I'm going to be talking about how do we identify research needs during the pre-ANDA process. Specifically, how early identification of research needs can ultimately help us to achieve our mutual objective of getting more high-quality generic products available on the market. This presentation reflects my views and should not be construed to represent FDA's views or policies. The goal of the presentation today is to discuss how GDUFA research has contributed towards the development of bioequivalence recommendations, and I'm going to be using topical dermatological dosage forms as an example. And then I'm going to talk about how identifications of research challenges and needs prior to the submission of an ANDA can be helpful towards the ultimate success of a product development program. And I'm going to be using topical gels, creams, foams as an example. So where are we at with these topical dermatological products? Historically, for these locally acting complex dosage forms, we have typically recommended comparative clinical endpoint studies and vasoconstrictor studies for corticosteroids. However, more recently, we have recommended efficient characterization-based approaches. The development of these efficient characterization-based BE approaches have been supported by research that we have conducted within the scope of the GDUFA Regulatory Science Research Program, and I'm going to be showing you a very small snapshot. In this example, we looked at metronidazole creams and gels. These products are known to be bioequivalent based on comparative clinical endpoint studies, and we did microstructural or Q3 characterization of these drug products. And what we saw was when the Q3 of the products were similar, the performance was similar, and it was distinctly different compared to products where the Q3 was different. And we saw this not only for the metronidazole drug products, but a whole host of drug products, which ultimately led to the development of efficient characterization-based approaches. Now, within the scope of these efficient characterization-based approaches, we have what we refer to as a modular and scalable approach to BE evaluation, where we are trying to develop a generic product that is very closely matched to the reference product, such that the performance of this generic product can be expected to be similar to what we would expect across different batches of the reference product itself. And we do this by recommending sameness in formulation, assessment of microstructural data or Q3 data, assessment of drug release, and as the complexity of the formulations increase as we go from single-phase gels to emulsions, we often also recommend IVPT or another biorelevant assay. And in some limited instances where there could be a potential systemic site of action, we may also recommend a PK study. And the types of studies we recommend for each of the product specific guidances typically depends on the complexity of the dosage form as well as the site and the mechanism of action of the specific drug product. Now that we have briefly discussed what these characterization-based approaches look like, let's talk briefly about what are some of the research challenges that we have identified. So the first example that I have for you is this topical gel. It is a gel which has microparticles or microsponges suspended in the gel. And we looked at these microparticles. These appear to be porous structures, and the drug may be present on the surface or within the pores. And you can read more about this research within our research report or within Dr. Zidan's presentation from SBIA last year. However, one key consideration that we identified is the location of the drug within these microparticles, either on the surface or within the core of the microparticles, has the potential to influence the drug release from these microparticles and ultimately the bioavailability from these drug products. So while for a single-phase topical dermatological gel, our research may indicate that recommending formulation sameness, Q3 characterization, and evaluation of drug release may be sufficient to mitigate the potential failure modes for PE. For these topical gels containing microparticles, we may need to consider additional studies. We may need to understand the materials that constitute these particles. We may need to assess the particle size distribution of the microparticles within the drug product. We may need to understand the morphology, the surface area, the porosity, the drug loading, the drug distribution, the localization of the drug, as well as the physical state of the drug on these microparticles to be able to ultimately understand what factors may influence drug release and bioavailability. The overall goal being we really need additional research to understand the additional complexity that's associated with these topical gels containing microparticles to be able to efficiently develop these characterization-based approaches for such products. The second example that I have for you today is for topical emulsions. And like I said on a previous slide, for these topical emulsions, as we get into biphasic systems, we typically believe that an IVPT study or another biorelevant assay may be needed to be able to mitigate all potential failure modes for PE for these complex dosage forms. Now I have the components listed from the product label for this drug product, clindamycin phosphate topical lotion. And we have often discussed that for these multiphasic systems, if the API, which appears to be hydrophilic in this example, is largely located within the continuous phase of the formulation, then do we really need an IVPT study? Well, we did some research led by Dr. Kandakar Alam from the Division of Quantitative Methods and Modeling. And in this small demonstration that I have on the slide here, we saw that as the formulation evaporates, the percentage dose of the drug in the continuous phase and the oil phase changes over time. And we know that the relative contribution of the drug in the oil phase and the aqueous phase may influence the bioavailability. So it is critical to utilize a study such as an IVPT to be able to adequately mitigate all potential failure modes for PE for a product which is a topical emulsion. And while we understand that there are implementation challenges with IVPT, we have discussed them in details at the workshop last year, we still think that this is an important component of a lot of characterization-based approaches where the reference product is an emulsion. And specifically for this clindamycin phosphate case, we had an additional challenge. Clindamycin phosphate has the potential to metabolize in the skin. So now we need to consider, do we monitor clindamycin in the receptor solution? Do we monitor clindamycin phosphate in the receptor solution? And how do we identify which analyte or analytes may be relevant for evaluation of PE when we use an IVPT study as a component of a characterization-based approach? So again, here is an example where we think if you are interested in developing such a product or similar products, early engagement and discussion between the industry and the agency may be beneficial towards the overall goals of the product development program. The last example that I have is topical foams. Historically, foams may qualify for a waiver of in vivo bioequivalence study requirements, and in those situations we recommend some characterization tests. For foams that may not qualify for the waiver but are corticosteroids, we may recommend BC studies, and for other foams we may recommend comparative clinical endpoint study. But what we do need to remember, and I'm just going to show you two foams with their respective compositions, and what we see here is some of the inactive ingredients that constitute these foams are complex: soybean oil, coconut oil, among other components. And what we need to consider is we are still at a stage where we are trying to understand the microstructure of these foams. We are trying to understand how these inactive ingredients may influence the microstructure and what the relevant Q3 properties may look like based on the complexity of this drug product. What Q3 do we need to monitor and compare? And lastly, how do we even conduct these performance tests? such as IVRT and IVPT. Do we use the collapsed foam? Do we use the foam as dispensed from the container closure system? The point being, we still need to do additional research to be able to develop efficient characterization-based approaches for these products. And in the meantime, if you are interested in developing such products, early engagement with the agency may be beneficial to ultimately move the products forward. In summary, I hope I was able to communicate to you that, like a lot of products we will be discussing today, these topical dermatological products are complex dosage forms. We have developed efficient characterization-based approaches for these products within the scope of the GADUFA research program. However, there are outstanding questions, and identifying these research needs during the pre-NDA process and early engagement between industry and the agency may be able to help us to develop strategies that can then be utilized to facilitate generic drug development. I would like to acknowledge and thank our entire team within the FDA, as well as all of our research collaborators and all of you for your contribution to this research program. And I look forward to your questions during the Q&A.